Tony, you're muted, Tony. You're frozen too.
Okay, no. Hopefully not. Can you get my voice clearly?
Yeah, now I can hear you. You're gonna want to share your screen.
Okay.
So just so you know, in the bottom middle of your screen,
Yeah, I see
just
it.
like Zoom.
Through this.
There you go.
Share
Very
screen.
well.
There we go. Share.
And while you're doing that, Tony, would you mind introducing yourself and your organization, telling us what you guys do?
Yes. And so, hello everybody. Thank you for joining us on the call today with Benzinga. Thank you, Benzinga, for hosting this. My name is Tony Tontat. I am the CFO of Kiromic Biopharma, and we are specialists in creating cell therapies for oncology, specifically solid tumors.
Fantastic. And were you able to get your screen share working there?
Okay, so screen share, here it comes, right? Here we go. And so you guys can see on here that this is our corporate presentation that we had done the IPO with, and our science is fairly complex. And so most of the time, I prefer to go through a generalist format in which I will offer you this following format. The technology module lasting approximately 4 or 5 minutes, the financial module lasting approximately 2 minutes, and then the catalyst module 2 minutes, and of course, most of the time spent on comparables thereafter. Does that make sense for you, Neil? Would that work?
That works for me. Thank you, Tony.
Right, so the Kiromic presentation, everybody can pull it directly from ir.kiromic.com. And so we can certainly share that, but you can see here it's fairly scientific, right? So I would like to offer the generalist format instead. Would that be okay, Neil?
I approve. Continue.
Okay, so hello everybody. So I'm gonna go ahead and launch into our technology module. Please know that there are liquid cancers, which are all the cancers that are going through your circulatory system, like leukemia. And then there are solid tumors. Kiromic is solid tumors. So we work on cancers that are essentially sitting on all of our organs. So that would be ovarian, prostate, lung, brain, kidney, so on and so forth. Next, for the treatment of cancer, there are what you see in the labs called small molecules created by humans and put into humans with significant side effects. What Kiromic does is called live cell therapy, otherwise known as CAR-T. In the live cell therapy, a T cell is extracted out of a donor, gene edited, and given to a patient. So that's what Kiromic does, a live cell therapy. And also within the live cell therapy, there is autologous format in which T cells are extracted from the patient, gene edited, and given back to the same patient. Here's a problem with that. When you go and use T cell from a sick patient, try to gene edit it, that T cell doesn't survive so well. So the next generation of live cell therapy is called allogeneic. You guys can see it directly on the screen. This is T cell directly from a donor. We take it out, we gene edit it, and then we give it back to the patient. Very similar to what you see in an organ transplant. And so within even the allogeneic live cell therapies, there are two different versions. The one-on-one version, which is very much like an organ transplant, or the version that Kiromic does. In the Kiromic version, the allogeneic, off the shelf, we take healthy T cells from a healthy donor, we gene edit it, and next we do what other people don't do. We expand that so that it can be used to treat up to 500 patients. And the typical dose is about 12 million cells. Okay, next we have biomarkers. And so most of you know biomarkers are probably proteins that are sticking out on the surface of the cell. And these biomarkers are used by scientists in order to differentiate cancer cells from healthy cells. That said, the Kiromic version, we use a proprietary artificial intelligence engine to be able to generate new biomarkers that improves our chances of differentiating healthy cells versus cancer cells. So that's it. That concludes our technology module. So I'm going to ask you if you have any questions. If you don't, I will go directly into the financial module. Neil, you have any questions?
So far, Actually, so there is one person here that's asking if you can comment on the status of the Rensselaer deal.
The Rensselaer deal, I don't know. We don't have that as a deal, sir.
I might have mispronounced it. Other than that, we don't have any questions right now. But guys, clearly Tony is open to taking questions. So make sure you're dropping those in chat, and we'll make sure they get over to him. So Tony, I think right now just keep going, and if we get some more, I might stop you and make sure you get those questions.
Okay, so on to the financial module. So in September 2012, the company was founded by Dr. Maurizio Cariva, Dr. Scott Delbeck, and Mr. Gianluca Rotino. We raised $23.5 million since the beginning of the company, and the company IPO on the 20th of October 2020. Since then, at the IPO, we sold 1.25 million shares, and we raised $15 million with a $13.24 million net raise. Our monthly burn is currently run at $650,000 to $750,000 monthly. Our cash runway is expected to extend to June to July 2021, so it's coming up very close. And without having to treat severe adverse events, it is likely that our cash runway could extend all the way to September to October 2021. Okay, so that's it. That's my financial module. And I'm sure you guys would have many questions upon our financing strategy going forward as we file the IND, getting the FDA approval, and then starting in human. So that's it. Let's go directly to the catalyst modules, right? In the catalyst module, in 2Q 2021, so we have approximately, 46 days until the end of the quarter to file our IND. Next, when we file our IND, it's likely that we will have 30 days until the FDA return and give us authorization to begin first in human. After we get that, we have announced that in 3Q 2021, we will be commencing our first in human dosing. And in 4Q 2021, we will be expecting the first in human safety and efficacy data from the trial. Okay, so do you have any questions for me, Neil?
Right now, everyone is really enjoying it, and they don't have any questions. So
okay, so I'll just go directly to our Kiromic presentation, and on page 27, I would jump right into the comparables. Right, so you guys can see here on the screen share, Kiromic truly is very unique because we are taking the intersection of technology and live cell therapy. So on the left side, you are seeing all the companies that are operating inside artificial intelligence and oncology. And then on the right side, you're seeing all the guys who don't have artificial intelligence for biomarkers, but they are using live cell therapy, very similar to Kiromic. So I'll just start with a few comparables on the left, right? So in Lantern, they have an excellent AI engine that is used to salvage clinical trials. So their AI engine combed through many clinical trials and they try to fish one out that they can save. Okay, and they run those trials again for a new indication or the same indication. But essentially, it's an AI engine for clinical trial salvage. Very, very successful, and I wish them best in that. And so the next one is BioXcel. In BioXcel, BioXcel, they have an AI engine that goes through many off-patented drugs. They take those drugs back and run the trials again for a new indication. So that said, BioXcel is an AI engine for extending patent life of old drugs. And then next, there are Black Diamond Therapeutics and Schrodinger. Both of them have AI engine value in the multi-billions, and they use their AI engine to do biomarkers very similar to what Kiromic does. But that said, we don't share our codes with them, and they don't share their codes with us. So they use their small molecules for therapeutics instead of using live cell therapy. So that said, you can see just on the AI component, Kiromic is priced very attractively. And so now let's take a look at the live cell therapy guys on the right side. All right, Juno and Kite, they have been acquired by Gilead and Bristol Myers respectively, just as they were finishing up phase 2 and going into their pivotal trial to get approval by the FDA. So they were acquired before they were approved. And then next you see Kodiak, value at nearly $350 million as of the latest. I can see that Kodiak had gone through their IND and got approval for the IND. And you can see that the value creation potential for Kiromic going from sub $100 million today all the way up to where Kodiak is. And Kodiak is also in the live cell therapy space, just like us. So if you think about Kodiak and Kiromic is approximately 45 to 60 days behind Kodiak. And then there is Fate Therapeutics, value in a multi-billion dollars. That's considered the bellwether of our space. They are doing solid tumors and CAR T, just like Kiromic. So if you look at Fate and you look at Kiromic, Fate is approximately 9 months to a year ahead of Kiromic where they are. So the potential for Kiromic to create value from a sub $100 million valuation is extremely high. So that's it, Neil. That concludes essentially all of my modules. And so you let me know what kind of questions that the generalist audience might have.
Yeah, so we have one. I'm not really sure what they mean, but I'm gonna give it a try. So have you seen your technology work on subjects before you start trials on humans, or is it still only on theoretical basis?
Oh, yeah. So, that CAR T back in September, October of 2014, it came to the forefront of science for the first time. The theory was that you can take a T cell directly from a human, you can gene edit it, and then you can give that back to the patient. And that is a lot more effective than giving a patient a small molecule completely from a laboratory. So in September, November 2014, that was a novel concept. As of today, that concept is alive, very, very much alive, with 2 marketed drugs on treating leukemia, and they are Yescarta and Kymriah. And so it's no longer a concept. What Kiromic is going to do that's different than the marketed drugs for CAR T is that we're going to go into solid tumors. Currently, all the CAR Ts, they only work in leukemic indications, meaning blood cancers. So solid tumors is still the predominant indications, right? If you have liquid and solids is like 90% of the market. So when Kiromic succeed in doing this, it's going to be a spectacular value creation.
That's remarkable. And how would you compare like your approach to this to your peers or competitors in the space?
Yeah, and so in the sense that when we are taking out T cells, there are different types of T cells. Some companies would go and take out T cells that are called NK cell. Others would use stem cells, and Kiromic is specifically using a subset of those T cell called gamma delta T cell. We had chosen that specifically because it is much easier to maintain in ex vivo expansion, meaning making clones of the cell, and then of course in gene editing, putting in information so that the gamma delta T cell know how to go and find a cancer so we are targeting. So those are the main differentiations.
And then do you have any operational or strategic advantages that you find compared to your peers?
Oh, yes. So when you think about, I'll just highlight a very specific thing for you, right? In other CAR Ts, the manufacturing of the cells, the cloning of the cell after the gene edit and extraction, it is very difficult. So cloning these cells, normally the CAR T company, they are not confident that they can do it consistently. So as such, they have to hire out contract manufacturers to settle the process. Kiromic is lucky in that we have the scientific knowledge in-house, and we also have GMP facility completely locked up so that we can manufacture all of these cells ourselves. Manufacturing, specifically, I'll use the word cloning, right? So you're trying to clone cell from a core block of cells.
Okay, so you're vertically integrated.
Yeah, correct. We're not allowing this process to go out-house.
Okay, yeah, great answers. Everyone's very, very pleased. From an investor perspective, if there's one thing that everyone should remember about Kiromic, what is that thing?
Yeah, so that we are solid tumors, allogenic, and off the shelf, meaning we can take a sample from a single patient, gene edit it, and expand it up to treat to 500 patients. So that is very unique because currently the whole entire market, they have to do a single donor, a single patient. That makes sense, right, Neil? If you think about it, right? If you are able to go and get one single good sample and you expand it for 500 different patients, you have a lot more consistency from batch to batch versus having multiple donors. So Kiromic is very, very special with that.
Absolutely remarkable, Tony. Incredible. Thank you so much for being here with us. I think that your scientific approach unto itself is absolutely fascinating. In addition to the investment opportunities you guys present, so thank you so much for being here with us.
Thank you, and we look forward to anyone who have any questions to go to our investor website, ir.kiromic.com, and post any comments, and we look forward to having further interactions with you and getting your answers on time. Okay, thank you.
Thank you, Tony. Thank you for being accessible.
Have a great weekend. Thank you, sir.
You too.